Our next speaker is Dr. Feng Zhang, who is a McGovern investigator and associate professor at the Departments of Brain and Cognitive Sciences and of Biological Engineering at MIT. Welcome, Feng.
Hi, good morning. First, I want to thank the Allen Foundation for supporting our work. And I want to give you an update on some of the work we've been doing on developing and mining novel microbial components to advance our ability to study cellular function, particularly for brain function. So you are probably familiar with the enzyme called Cas9. Cas9 is the RNA-guided DNA nuclease found from the microbial immune system called CRISPR. There are two main ways that Cas9 has been developed and applied so far. The first is using Cas9 as a nuclease for gene editing. By programming the enzyme Cas9 with a specific RNA guide that targets a specific sequence on the cellular genome, this enzyme can make double-strand breaks using these two catalytic domains to then trigger DNA repair processes. It can either fix DNA through non-homologous gene joining. It's a very useful method for disrupting genes because it forms indels. Or you can provide a synthetic DNA template to perform DNA repair. But the same enzyme can also get mutated, so you can get rid of the RUF-C and HNH catalytic residues and turn it into an RNA-guided DNA binding domain like this. And so using this, you can bring transcription activators to turn genes on. You can bring HDAC modulator to be able to turn genes off. Or you can even bring cytidine deaminases to alter specific DNA bases, or use GFP to visualize the dynamics of chromatin as cells are undergoing different types of morphogenesis. So this is what has been done to develop Cas9 into a wide array of different tools, forming a toolbox for perturbing cells at the DNA level. One of the questions that we were interested in is looking beyond Cas9 and asking the question, besides Cas9 in these CRISPR systems, are there other enzymes that can allow us to expand the number of operations that we can perform inside of a cell? So in order to move forward, let's look at the CRISPR diversity known back in the year 2011. And so each one of these sets of genes denote one type of CRISPR system. And so CRISPR, in fact, represents many, many different bacterial immune systems. Right over here is the Cas9-based system, and you can see that this is really the simplest relative to the other CRISPR systems. And simplicity is nice, especially for engineering the system to perform different applications. So all these other systems use many different components to perform the function that Cas9 is able to do as just a single protein. And so based on that division, we can reclassify CRISPR systems into two different classes. And so there's the class 1 CRISPR systems, which use several different proteins that form a complex to mediate the immunity effect. And then the class 2 CRISPR systems include Cas9, use just a single protein working with the RNA guide to be able to carry out defense. So back in 2011, there were many different class 1 systems known. So these are all class 1. These are also class 1. And the only known class 2 system was Cas9. And so we wondered if there are many class 1 systems known, could there also be multiple class 2 systems? And if there are, what do they do, and can we also develop them for interesting molecular manipulation applications? So we set out on a computational expedition. We collaborated with Eugene Koonin, and we set out this pipeline to be able to discover novel class 2 CRISPR systems. The way this works takes advantage of the very idea that CRISPR systems are bacterial. That means genes that are related to the same function, genes belonging to the same CRISPR systems, are most likely encoded within the same operon. And so nearby genes sort of suggest the function of their neighbors. And so what we did is we chose one of the most well-conserved CRISPR proteins called Cas1. And we use it to blast against the bacterial sequence database to see what are all the different contigs that carry Cas1. Once we found Cas1, then we can start to annotate all of the open reading frames that are nearby. So each one of these contigs has Cas1, but we can also figure out what are the nearby genes. For each one of these open reading frames, we can then blast it to see is it homologous to a gene of known function, or is it really just a protein that we don't know what it does. And so we naturally focus on these genes of unknown function, because if we only look at the genes that are homologous to genes of known function, we won't find a new CRISPR effector. And so based on that, we then found a list of different genes of unknown function. And then we apply two criteria to this. And the first criteria is for each one of these genes of unknown function, if you blast it, do you get a whole family of them? If that's true, then you know that this is highly conserved. And so evolutionarily is probably doing something that's important. And second, for all of these family members that belong to the same family, are most members often found next to other CRISPR proteins or CRISPR RNA sequences? And if that's true, then we know it's not only evolutionarily important and conserved, but it's also most likely a conserved CRISPR protein. And so based on that, that's how we ended up with a list of different candidate novel class 2 CRISPR effectors. So this is what we know to date based on the sequence database that exists. Of course, as we sequence more and more bacteria, it's very likely that we'll find additional types of CRISPR systems. So there are really three class 2 CRISPR systems. There's a Cas9-based system, and then there's a new DNA targeting system that's based on Cas12. You can also use these for gene editing. And then the really interesting one are the third type, which is called Cas13. It uses RNA guides to recognize RNA and then carry out RNA modification functions. So we've done a lot of work with Cas9 and Cas12, but I thought for today I'll just tell you more about Cas13 because it has a number of interesting applications for both perturbing cells, but also for developing novel diagnostics and therapeutic uses. So when we find a new CRISPR system, like a Cas13 system, the first thing we do is we try to put it into a genetically tractable host. So Cas13 is found in a bacteria called Leptotrichia shaki. And it's a bacteria that's hard to grow and it's also difficult to genetically modify. So rather than working in that native host, what we do is we synthesize the entire CRISPR system from that bacteria, transplant it into E. coli, and then now we have a system that's much easier to experimentally modify. And by doing RNA sequencing, we found that the CRISPR array, the repeats that are the signature elements of any CRISPR system, is expressed. And furthermore, these guide sequences shown as diamonds are expressed into individual mature CRISPR guide RNAs. So this is a very good sign. It suggests that this system is produced in E. coli, and it's also likely to be functional because the RNAs are matured into the right form to mediate immunity. So based on analysis of the Cas13 sequence, we hypothesize that it targets RNA because we find these motifs that are only found in RNases or bacterial toxins. And so based on the hypothesis, we then designed a screening assay. We began with a phage that's called MS2. It only exists in RNA form. And it's got a small genome, only 3,800 bases. So we can design guide sequences that cover from the beginning to the end of this entire virus genome. And so computationally, you can design all of these different guides. And then you can synthesize it very easily using DNA chip synthesis. So on a DNA chip, you can synthesize DNA oligos corresponding to every single one of these guide RNA sequences. So now we're ready to do the experiment. We clone these spacers into plasmids. Each one of these plasmids carries the full Cas13 system along with one of these spacer sequences. So then we can transform this library of plasmid into E. coli, and then we now have a library of cells. What's unique about different cells is that each one carries a different spacer targeting a different spot on the virus. And so now, if you challenge this library of cells with the MS2 virus, you can ask questions like, do any of them survive? If they survive, then we know that this system is active and it provided immunity and protected the cell from that virus. And second, by sequencing the surviving cells and analyzing the sequences, the guide sequences that are left over in the cells that survived, we can begin to understand what type of targeting motifs or what type of targeting rules are necessary for this enzyme to work. So it turned out that the targeting rule is quite simple. If this is the RNA that we're trying to target, and this is the site that we're targeting, then it only needs to be flanked by a single base. And that can be either a C or A or a U. And so this makes targeting within any RNA molecule fairly easy. And we then purified this enzyme just to test biochemically whether or not this targeting rule is robust. And so what we did is we have a piece of RNA, and we designed different guide sequences that targeted this RNA based on complementarity. And then the target site is designed to be flanked by different RNA bases. So if you look at an experiment, this is the RNA we're targeting. These are all of the different sites that we targeted within this RNA. You see that any guide sequence that's flanked by a C, a A, or a U successfully cleave the input RNA into different fragments. But when you have a G, you don't end up with cleavage. And so this confirms that we have this single base requirement for Cas13 mediated RNA cleavage. So what is also interesting, in addition to cleaving the recognized RNA, we found that it also has a second RNA activity that we call collateral activity. So the way this works is, if you look at this experiment, we'll have a guide RNA and we'll have a target RNA, but this target RNA has no homology to the guide, so it's not recognized by the guide RNA. And if you put Cas13 in this reaction, what you will find is that nothing gets cleaved because this RNA doesn't recognize the target RNA, and you just end up with no cleavage. But if the target RNA is present, then we see something that's quite interesting. Now, we have the target RNA, and it's base paired with the guide, and so this target RNA will certainly get cleaved. That's what we saw in the gel before. But what's interesting is that these collateral RNAs, even though it's not recognizable by the guide RNA, when in the presence of Cas13, they will also get cleaved into different fragments. And so this suggests that Cas13, upon recognition of the target based on guide RNA complementarity, is switched into this non-specific promiscuous state, and it can go and cleave other RNA molecules that are in the vicinity of the enzyme. So what does this all mean in the context of the bacterial immune system? So we think that in addition to providing the standard form of immunity, which is that when you have an infection, it got into the bacteria, and then the Cas13 acting as a sensor recognizes this infection, and then immunity will destroy that virus, cleave the RNA, circumvent replication of the virus. But a second mode of defense is where if the virus escapes the sensor and begin to replicate and make many copies of the virus in that cell, now the population is at risk, because if the virus escaped, all the replicated viruses escaped, then they can infect many more cells, and the infection is now not under control anymore. So rather than letting the virus escape the cell, what Cas13 can do through this collateral cleavage is that it can mediate programmed cell death. So each molecule recognizes a single replicated copy of the virus, and then when the virus is amplified, you have many, many molecules of Cas13 that can go around a cell and cleave enough RNA to then destroy the cell. And so that destroys the cell, the infected cell, but protects the overall population. So that's what we think Cas13 might be able to do in addition to providing the initial, the basic form of immunity. But what is this all good for? So it turns out that we can take advantage of the collateral activity to develop a easy, fast, and also sensitive diagnostics assay. So just this morning, the WHO Director General called an emergency meeting to assess the Ebola infection, the Ebola pandemic that's been happening in the Republic of Congo. And so for diseases like this, we really need to have tests that can be performed in the field in an efficient and also specific way, and also at a very low cost. And so using the collateral system, we can now use a method that we developed called Sherlock to detect viruses like Ebola or Zika or even other bacterial pathogens. So the way this works is we take advantage of collateral activity to cleave a reporter RNA in the same reaction. So here, this blue glob is the Cas13 enzyme. And if we're trying to detect Zika sequences in a biological sample, blood, urine, or saliva, we can program this enzyme with a guide RNA that's been designed to recognize the sequence on the Zika virus. But in addition to providing the guide RNA, we can also put in a reporter RNA that has two moieties on the two ends. And this can either be a biotin or be a FAM a molecule on the other end. So if the Zika virus is present, then the enzyme and guide will recognize the Zika virus, cleave the Zika RNA, but at the same time activate collateral activity and then cleave these reporter RNAs to separate a biotin away from FAM. This makes it possible to develop a simple paper strip-based test, very similar to what you might see in a pregnancy test on paper strip. And so the way it works is that we have a paper strip, and there are two lines that can capture the two different moieties on this reporter. So the first line captures biotin, is streptavidin, and then the second line is the antibody that can bind to the FAM molecule. And so if the virus is not present and the reporter is intact, when you float the reaction on this paper strip, everything will get captured by the streptavidin line. And that's because biotin and streptavidin are linked by this reporter RNA. But if the cleavage happened, meaning that the virus is present in the reaction, you end up with two lines. You get streptavidin capturing biotin, but FAM can keep flowing, and they can get captured by the antibody capture line. And so this is what allows us to then be able to very easily detect whether or not a particular pathogen is present in a biological sample. So this is what it actually looks like. This is one of our earlier experiments. And in here, we found that with different concentrations of Zika virus, we find that you can get to about 200 attomolar. And so this is about 100 molecules of the virus per microliter biological sample. And so with further optimization now, we have gotten this down to where we can approach the single molecule per microliter level using this paper-based test. So this is all for detecting a single thing, but sometimes you may want to develop a panel if you want to check to see are there multiple different pathogens and which one of them is present in this patient sample. And for that, we went back to the drawing board looking at this gel to then think about how do we develop a multiplex-based detection assay. And so if you look at this experiment, this is the guide RNA, this is the target RNA, and you have many different guide RNAs targeting different positions. But it doesn't matter where these guide RNAs are targeting on RNA, you end up with exactly the same cleavage pattern. And so what that suggests is that it's not cleaving in a recognition site specific way, but rather maybe motif or secondary structure specific way. And so we thought maybe if that's the case, different Cas13 family members might prefer different motifs. And therefore, we can find many of them that cleave in different orthogonal motifs to be able to multiplex it. So then we set up this experiment where we have different Cas13s, and we provided different reporters that are separated by different sequences linking the two moieties. In this case, it's the fluorophore and the quencher. And so what we found is that different Cas13s can indeed have specific cleavage preferences. LWA cleaves AU sequences only, but not UC, AC, or GA. And then CCA cleaves UC only and not the other ones. And so they are orthogonal to each other. And by studying the whole family of Cas13 enzymes, you can find a whole set of them that can cleave different sequences. So then using this, we can develop a multiplex assay. And here we're doing a three-plex assay where we're trying to detect Zika, dengue, or synthetic RNA. And we'll have different enzymes, different guide RNAs, and also their corresponding reporters. So what you find in the same biological assay is that when nothing is present, you don't get any signal. If one of the three species is present, you get one of the three. If you have two of them out of three, then you get two of them reporting signals. And if you have all three, you get all three signals. And so you can multiplex them and also read out the result in individual channels that don't crosstalk with each other. So this is one way to use Cas13, but in addition, because it's RNA targeting, we can also use it to edit RNA. So we can bring an enzyme that can convert adenosine into inosine to be able to alter the protein that's translated from a given messenger RNA. And so this is a schematic of how this system works. We can take Cas13, deactivate it so that it only binds to RNA, and bring ADAR, which is the enzyme that converts adenosine to inosine, to a specific transcript in the cell that we're trying to change. And then by giving it a guide RNA, we can specify a specific base by mispairing that adenosine we're trying to edit with the cytosine. And then so when it forms a complex, the mismatch base gets converted from adenosine to inosine. So inosine is interpreted by the translation machinery as a guanosine, and so you end up with a amino acid change for the protein product that's produced. So this, as well as the detection system, are just two ways that we can use these new bacterial enzymes to be able to further study biology and also try to improve human health. But one of the things that we're really excited going forward is to ask a broader question. So besides CRISPR-based immune systems, are there other immune systems too? So these are 4 different examples of immune systems. There's the CRISPR RNA-guided nucleases, antibodies from our own species. There are another form of antibody called VLRs from jawless vertebrates. And then the tail proteins is yet another immune molecule that's produced by bacteria. And so these are all systems where they rapidly evolve and are adapted by nature to be able to recognize different substrates to be able to allow the bacterial species to survive in the face of adversity. And so one of the questions we have is, are there more programmable systems besides these? Because they're likely to be new adaptive systems that we may be able to harness and also reprogram to be able to further study biology. I'll just briefly show you 2 examples. This is a protease. It's got a repeat domain that's kind of like the tail systems. And these repeats are mostly conserved, but carry hypervariable regions that might confer substrate specificity. And then this is another system where there are these clusters of highly repeated proteins. They are pretty much identical to each other except for a fixed window of high diversity. And so by looking at this, you can see that the sequences are very diversified, and we're trying to figure out what this might be doing in the context of bacterial immunity. So that's all to say that if you look at the diversity of bacterial sequences, which has grown quite dramatically over the past decade, now we have 140,000 complete bacterial genomes and many more in the metagenomic sequences. So this wealth of data really is worth mining, and we'll probably find many interesting biological processes, and some of them will be likely useful. So finally, I just want to acknowledge my team at Broad and MIT, our collaborators, and also the funding agency, and most importantly, the Allen Foundation, where supported some of this work. Thank you very much.
We have time for one question.
Yeah.
That was really great. Just thinking actually about Jeff's talk earlier, and the localization specific RNAs, because what you'd love to do in that context is not hit the gene, but hit the RNA to demonstrate that localization has a function.
Right.
Can you modify Cas13 in a way, I'm sure you're screening for this, that its collateral activity is inactivated so that you could get it to only cut the target RNA and not other RNAs? I mean, presumably you can screen for that with your double probe, right?
Right. Good question. So that's certainly doable. The way to do that is not by mutating Cas13 alone, because the domains that's responsible for both target cleavage and also collateral cleavage are the same domains. So you can inactivate it, but bring a separate RNase along with it to be able to destroy the recognized RNA. And so, for example, the pin domain is the RNase that we can use to cleave RNA.
So you don't know anything about how, what it is about the recognition of the target RNA that converts it in a way that allows it to become now a collateral cleaver. What's the confirmation?
Yeah, that's a really good question. So we now know a little bit more about that. We now have a crystal structure for Cas13 bound to the target. And so what is happening is when the guide RNA recognizes the target RNA, it triggers a conformational change and switches the enzyme into this active RNA state. And it's that activated RNA state that goes and cleaves both the target RNA, basically motifs that are at a distance from the recognition site, as well as any other RNA that's in the neighborhood. Yeah.
Okay. Thank
Thank
you
you.
so much.